Okay, good morning, all. I hope you have revised all your previous sessions and also the questions which we have discussed last class. Sorry. So, there are any doubts with the question bank what we have discussed
No, sir.
previous session? No.
No, sir.
Okay. So we have another one topic which is left in the unit 1. Okay. And that is nothing but genome annotation. I have already started that in the previous class, just I gave an introduction. So now we'll continue with that genome annotation. Okay. Okay. So genome annotation is again I'll start from sessions. Okay. So genome annotation we are going to cover introduction, object possession, what is genome annotation summary. Okay, and we have seen that genome annotation is nothing but gene prediction and functional assignment, which is done in 2 steps. And this is used to analyze all the biological features which are present in the sequence, whatever we have assembled previously. Okay, so previously we have learned that we can do something. From that we can do this. Okay, so genome annotation is nothing but first step is to predict gene, and second step is to assign a gene. At the same time, annotate all other parts. Okay, all other elements of the genome. So you can see here, there actually when you go the conventional textbook, you look at the conventional textbook, or when you go through the papers or anything, genome annotation is actually defined like this. But practically, genome annotation, we have to say there are 3 possible steps. First one is gene prediction, second one is functional assignment, and third one is again annotating the other elements of the genome, which are not genes. Okay, like your transposons, retrotransposons, your tandem repeats, your pseudogenes, your promoters, all others will come in this. Okay, so this is genome annotation, and we'll learn this in detail when we are going to study about the genes, CPG islands, promoters, functions of the genes, etc. in the 3rd unit. Okay, this is just an outline of what is actually genome annotation. Okay, so the object of today's session is, as usual, to discuss in detail about genome annotation. Okay, so the first aspect what we do here is we employ certain tools or a combination of methods. So we have 2 methods here: theoretical prediction and experimental verification. First, theoretically, we should know what is a gene. Okay, and then only you can experimentally verify it. Okay, so any gene annotation, so gene prediction is actually done with gene annotation. Okay, and that is actually first you have to predict a gene and then verify whether that is a gene or not. So this uses 2 things: theoretical prediction and experimental verification. Okay, so generally the tools which are already used in large for scanning a bacterial gene is nothing but gene scan or gene sticks. Okay, and the method which is used here is ab initio method or homology method. Okay, so there are 2 methods here: ab initio method or homology method, which are theoretical predictions, and the tools which are developed on this are nothing but gene scan and FGH. Okay, in bacteria. And coming to eukaryotes, the ab initio method also looks at the exon production. And there are several tools which are available for us. Okay, and that is gene mark, that is known as gene mark eukaryote. Okay, now once these tools are used for theoretical prediction, this is a theoretical prediction. Whatever in mathematics we do, they are actually known as theoretical prediction. That means we first have an idea. For that idea, we get a proof that this is there, and this tells that there is a chance that this can happen. So theoretically, what we have done, we have identified that these elements of the gene are there in your sequence, and then you verify it experimentally. Okay, experimentally. So that can be done by predicting using BLAST. I think you've heard about BLAST. And what we do in BLAST, we take a sequence, we search against a database, that is sequence database here, and we see whether there is any similar sequence or not. If any similar sequence is there, that means that sequence is near to our gene, and so that gene is already existing in the nature. So then you try to amplify it via PCR, clone it in the lab, and then sequence it with Sander's method. We follow all these steps. Okay, and then we say this gene is present. Okay, so once this gives an idea that theoretically the gene is predicted using gene scan, things have, and that prediction is verified by BLAST against a sequence database, and then you can use a wet lab verification, that is experimental verification. You can make a PCR reaction to completely amplify the gene, and then you can get that whatever amplified product, you can subject it to sequencing by Sankers and then get the sequence. Okay, in this way, we can know that this gene is actually existing in an organism you have identified. Okay, so that is experimental verification. Now, how do you know that whether it is functionally expressing it or not? So I said that presence of gene, it is different, and expressing it is different. So I already gave you an example that uh, I mean, a person who's standing there like that, still or sitting there, it's just there. That's all. We don't know whether it's doing a job or not. But when a person is actually involved in work only practically, then we say that he is actually working there. So presence of the person at that moment and working of the person is a difference there for, I mean, saying that a gene is present and a gene is functionally present. Okay, so for identifying the gene which is functionally present, we have two methods here. First one is experimentally determined cDNA, okay, and EST. So already we have several methods. So what we do is for certain conditions we express the we look at the expression of the gene via cDNA. cDNA is nothing but the complementary DNA, complementary deoxyribonucleic acid. Okay, that cDNAs are the full length of the sequences. So the full length of the gene are actually available here as in the form of cDNAs. But when the same gene which is expressed, but it is present in the form of short fragments, maximum of 500 base pairs. Okay, those genes are expressed, but they are only of maximum of 500 base pairs. Then they are known as ESTs. That means expressed sequence tags. Okay, expressed sequence tags. When this is done, then you have functionally verified genes. Okay, so we have two types of databases which are available. cDNAs in the form of full length which are expressed, ESTs in the short form which are expressed for the genes. So what we can do is again we can make online verification of search via BLAST. Okay, so we BLAST these genes, whatever gene you have identified, against the cDNA database or against the EST database. Okay, we can make it in two ways: cDNA database and EST database. So when you do it either with the cDNA database or EST database, we have an evidence theoretically saying that this gene is expressed and this gene is available already online. Okay, that means there is a chance that this gene is present and this gene is expressed. So the tools which are used to identify these genes are nothing but via cDNA or EST are nothing but gene-wise, SPIDey, SIM4, and EST2 genome. Okay, these are the four tools which are. So how do you do all this procedure? That means theoretically predicting the gene, then verifying the experimentally using BLAST against the cDNA database or against the ESC. Yes, nothing but all this data is actually manually curated, right? So what we do is, whenever a person says that this gene is identified, they have to deposit in a certain databases like NCPI, DDBJ, or EMBL. So when you submit there, that will be manually checked and curated. That means we store it, all this data. So human curators are there to check whether this gene is accurate or not, data is correct or not. So after this verification is done, that data will be available either at the NCBI or at the DDPG or at EMPL. So when this data is available at these places, then this data will be make us available, and we can make a similar search with the cDNA or ESC databases. Okay, so this is how a gene is prepared theoretically, and a gene is verified at cDNA and ESC. Okay, and the tools which are used here to do that are GeneWise, Spidey, Simfo, and ESC. Okay, and this is how we try to predict a gene. Okay, this is how we try to predict a gene. Okay, so now we said that a gene is present and you have done several steps to predict the gene. Okay, so first you have got the gene theoretically identified, then you have identified whether this expression version is there or not, either in cDNA form or in EST form, and then you have confirmed it. But once you confirmed it, that gene is present, we should know what does the gene do. Okay, the first thing is we have predicted the gene, but second thing is we should know what the gene is actually doing there. So when you know what the gene is actually doing, then only you can say that this gene is so and so present and having so and so function. Okay, so the functional assignment here is done in different ways here. The first immediate step what we can do when you want to assign a function to a gene is just simply use a homology method, that is search this gene against a database. Okay, so you submit a nucleotide sequence, but you ask for a protein product. Okay, so you have seen certain BLAST variants there. BLAST variants. In that, one of the important variants is that you submit your sequence of nucleotide, but you search for similar protein sequences which are there. Okay, so can anyone tell what is that BLAST variant there, which is actually used for identifying protein, but you submit a nucleotide sequence? Anyone in the class? Yes. Anyone? Balaji? Balaji? Are you there in the class? Gunadeepak?
Sir,
What is the variant of BLAST which can be used to identify the protein sequence, but there we submit nucleotide sequence, not protein sequence? Can anyone tell?
sir, blast N, sir.
BLAST?
N, sir.
N.
Yes, sir.
BLAST as N is generally used for identifying similar sequences.
Blast X, sir.
BLAST
X.
X. Are you sure?
Yes, sir. Like it was long ago, so I don't
No, you
remember.
have to remember, because each and every class what you have learned previously is a
Yes, sir.
I mean, it will be an addition. That knowledge is actually required for when you go to the next semester papers.
I think it's Blast X, sir.
You do one thing. Okay, search this question again in the break time when I give you. And
Sir,
tell me as soon as the break is over.
sir.
Yeah, Malav.
Sir, my earphones got stuck, sir. Like
Okay,
they
okay.
didn't unmute. I removed them, sir. No.
You don't know the answer?
No, sir, I don't know the answer.
Okay, just verify it and tell me.
Okay, sir.
Okay, I'll wait for the answer. Okay, so
Sir,
yeah,
is it E-Blast X?
no, there is no E blast something like that. There is blast X, blast and blast
T,
B,
sir, T. E-Blast X.
T, blast X. Yes. Okay, you give a nucleotide sequence, it gets translated. Okay, and then it gives you the protein sequence. That is a simple method, but you should know that we will be using that. Okay, so are you clear with this now? Don't they pack others the class?
Yes, sir.
It is class T,
Yes, sir.
actually, class T. So what does it do is it translates the sequence initially, and then after translating, it gives you it retrieves the protein sequence if any sequence is there. And then based on that, it will say that this is the function of the gene. That is a very simple method. Okay, but the general orientation is that you not translate it directly using the BLAST, but we have to follow a certain method. I think you have done the practical also for that. Do you have any idea what is the practical you have performed already? The last class I asked you, you said that there are 2 practicals you have performed already in genomics.
So we
Anyone?
did blast.
Hmm?
We did not perform blast yet, sir.
No, it's not
We
blast.
performed it in third semester, sir.
No, what experiments you have completed in this semester? I'm asking you. Oh,
Sir, ORF finder.
yes,
Then domain, like we search for domains in a sequence.
yes. Then
Then there's an experiment called acquaintance with like genome databases, sir.
okay, these are the 3 you have performed in this
Yes,
lab.
sir.
Okay, so now,
Another one we did, sir, string, regarding string.
okay, so all these 3, whatever you have learned, among them, 2 are very important to you now at this moment for this class. Okay, open reading frames. Open reading frames. That is the first important one while you assign a function for a gene. Okay, so open reading frame is very important, and we'll see how it is used actually for us. Okay, so when you were taking a sequence, how many open reading frames did you get? How many open reading frames do you get in the practical version?
Sir, it depended on the size of like sequencer, length of sequence.
Length of the sequence. But how many did you get?
Sir, I got 2, sir.
You got 2. Okay, anyone? Any other number?
13 for me, sir.
8, sir.
Okay. So what do you mean by open reading frame here? Then? So you have done the practice. So by this time you should know what is open reading frame. Now what is open reading frame? Yes. The most important version of this. What is open reading frame? Varshita? Varshita? Padmini? Varshita and Padmini. Yes, respond. What is open reading frame?
Sir, is it a part where protein sequences or amino acid sequences are produced?
It's the part in which
Protein or amino acid sequence are translated.
are translated.
Sir, and it has a stop codon and start codon. Like,
Yes. So open reading frame, the name itself will tell you that a sequence where a frame can be opened and a frame can be closed. When does a frame open? A frame opens when there is a start codon and the frame closes when there is a stop codon. Okay? A frame opens when there is a start codon and a okay. So how many open reading frames are possible theoretically when you have a sequence with you? Yes. Anyone in the class? Okay.
So like
Yes, tell me.
6, sir.
6 open reading frames are possible. So how are they possible is, I'll show you here. Assume that this is your sequence. Okay. And in this sequence, the first open reading frame, ORF1, will start from here. Okay. From A. So the first 3, you'll get one amino acid. Your next 3, you'll get second amino acid. And third one will be left. Okay. Assume that there is CCG. Okay. So this is the third amino acid. This is frame 1. Okay. Now look at frame 2 now. Frame 2. So frame 2 starts from here, from second amino acid. Okay. So last there are 2 amino acids which are left. This is ORF2. Now coming here, how many are possible with third one? So third one starts with your third amino acid, third nucleotide. So there is amino acid 1, and from GGC, amino acid 2, and 1 nucleotide is left. Okay, so this is in one direction, 5 prime to 3 prime. Assume the same thing happens in reverse direction again. So there are, in reverse, you look at this. This is ORF1, and if you go in the reverse direction, so the orientation is this for ORF1, and the orientation will be this direction. Okay, orientation has changed. So are you able to understand how we are getting these open reading frames and how your software recognizes these open reading frames?
Yes, sir.
Is it clear to all?
Sir.
Yes, sir.
Any doubts here? Any doubts?
Sir, if it's a longer sequence, will it be 6 or the number will increase, sir?
No, see, what I'm talking is these are the minimum possible ORFs. Okay. If your open reading frame is there when it starts with the new start codon and ends with the stop codon. If your start codon or stop codon are not there, then you will get a variation. So that is a variation what you got previously. I mean, some people said you got 2, some people said you got 18, some said you got 12. You got variation, isn't it?
Yes, sir.
That variation is that this is actually the minimum possible theoretical ORF, but the variation is there because maybe some of the sequences are not having start codons and some of them are not having stop codons like this. Okay, this should be a start codon and this should be a stop codon, and reverse, this should be a start and this should be a stop. When these are not there, then your ORFs will not come. So that is how the variation is there. Another possible variation is that if a sequence is long, have you heard of nested genes? Have you heard about nested genes? Anyone? Anyone in the class? At least the term? No?
The
Yes
term,
or
sir.
no?
Yes, sir.
Yes?
Yes, sir.
Yes. Then what do you mean by that? If yes. Okay. So if a gene is there and the gene is a longer sequence, which is 1,000 base pairs, and another gene is there in that gene, which is of 500 base pairs. So gene is there within a gene. Okay. So I'll erase you this and I'll give you what is actually a. I think get it. Okay, so when you have a gene like this is your long gene one. Okay, so when you have a gene like this is gene 1. Okay, and you have another gene in this part. This is gene 2. This is known as nested gene. Okay, this is known as nested gene. So this nested gene can give you this variation. So those who all who got more than six. How many of you got more than 6? Can you raise your hands? How many of you got more than 6 in your experiment when you done ORF with ORF? Only one? Rest? So 4. That's all. Rest of you got less than 6 then? So those whoever got more than 6 here, the reason for them is that your gene is actually that area segment, whatever you have searched, they're actually nested genes. And so you got more open reading frames. So generally with one gene, we can get 6 open reading frames. So when you have 2 genes, you can have minimum 12. If you have 3 genes within the same gene, then you can have 18, which is possible. So 6, 6, 6. Okay. So this is how you will get more open reading frames. Okay. So once you got this open reading frames, whatever 6 I showed you or whatever number you got, what did you do in the next step? Balaji, after getting the open reading frame, what did you do?
Sir, we selected, like after getting the list of the ORFs, we selected a single ORF and then we looked into it, sir.
You selected the?
We selected a single ORF and we looked into it.
You selected only the single ORF or you have gone with all, you have blasted all of them?
No, sir, we didn't do blast for it.
Each and every. No. So
No,
the thing
sir.
here is what you should do is you should blast each and every open reading frame. Okay. You should blast each and every open reading frame. Take one open reading frame, then blast it. Okay. And then you will understand whether that is really an open reading frame or not. And
Okay.
then among them, one will be actually your gene. Okay. One will be actually your gene in prokaryotes. But coming to eukaryotes, there can be many variants. There can be many variants for the gene. So you might have seen, for example, you might have seen online that beta tubulin gene is present. Alpha tubulin, beta tubulin, gamma tubulin, or
Yes,
your
sir.
hemoglobin genes. Globin, alpha globin, beta globin,
Yes, sir.
okay, delta globin. Why? How you are getting this? Because the open reading frames, when with one gene, when there are different open reading frames, that means the same gene is coded with a little bit of variation for the gene. Okay, there you will get the variance. So this open reading frame can give you the variance if it is in eukaryotes, but if it is in prokaryotes, you have only a single gene. So you have to blast or search against all genes. So once you got the open reading frame, you might have taken a sequence, you have found out the open reading frame, and then you might have got the blasted and got the function of the open reading frame. That is what you have done with open reading frames in your practical, isn't it? Apart from this, did you do anything with open reading frames?
No, sir.
No? Okay.
No, sir.
Now the second question is, if you get a sequence, result with the blast with the open reading frame, is fine. If you don't get any similar sequence when you blasted it for the open reading frame, then what you can do? Does that mean that there is no function for that open reading frame, what are you predicted? Or you should look in another way saying that we did not get a positive result, but there is a chance that that open reading frame may have a function. In what way you have to look if you don't get the result?
Sir, the second way, sir. Like it may have a function.
Okay. So the second option you have to look. So once we blasted the open reading frame, and if you don't get any result with whatever open reading frame when you translate it, that means it does not end there. You have to think in another possible way, that there is a sequence which is there, and as there is a sequence, there can be a function, and that function we are not knowing it at this moment, but we can know the function by predicting it. So the second way what we do is with the open reading frame is we go with domain and motive search. Okay, we go with domain and motive search. I think you people also perform this expert, searching for domains, searching for motives, isn't it? Done? Have you done this?
Sir, only domain, sir, not motive.
Okay, so both are similar. Okay, both are similar. So the second option what we have here is when you have a open reading frame, what you do here is to just make a homology search and identify the function of the open. If you have not identified it, then you'll go for a domain or motive search. Domain search you have done using your conserved domain database, isn't it? At the same time, we have motive databases, okay, which you search it. In the general, you might have heard about the PFAM database and InterPro, okay, which are actually a database compiled of these motifs and domains, protein motifs and domains, and they give you the function of the. So PFAM is nothing but protein family, and InterPro is nothing but interrelated for proteins data. Okay, and these are the two tools which are mostly used to rely whatever functional predictions you have done. Okay, so this is how the first step what you do is in the open reading frame is first identify the open reading frame, then search for blast. If you don't get any result for blast, then we go for domain search or for the motive search. Domain you have done conserved domain, and motives we have a motive database. Apart from this, you can use PFAM or EnterPro based on motive and domain searches. Okay, this is how you assign a function for your gene. So what we have done, we have taken a gene, we have predicted theoretically, then we made a homological search. Then after that, you search that sequence against the cDNA database or an ESG database, and after finishing that, we confirm that is a gene, and then based on the gene, then we go for a functional assignment. So what we do here is we first predict the open reading frame, and then we go for homology search using BLAST. If something is not predicted using homology search, and then we go with the domain or protein motive database, that is PFAM or InterPro or conserved domains or motive database. Okay, in this way, you try to do your gene prediction and functional assignment. This is a traditional way of doing it. Okay, this is your traditional way of assigning a gene or assigning a function for the gene. Okay, and there are many more methods which have evolved based on this traditional method. This is the most simple, best method what we have, and there are more traditional methods. So we'll take a 5 minutes break, and after that we'll come and see what are the methods which are evolved from this traditional method and how we will do genome annotation at this moment. So any doubts here till here for gene prediction and functional assignment of gene? Yes or no?
No, sir.
No doubts? Okay, we'll meet back on around 9:45. Okay, and we'll continue with the next one. Okay then. Okay, so once we have used this traditional method, so what happens if we don't even determine the function of the protein using your open reading from and BLAST and domain database and proteins. What is the other way? So the other way people started identifying the function or assigning the function to the genes is using gene ontology. Okay, gene ontology. Gene ontology is actually but a language which is actually a natural language processing. So any natural language, with what we call, they have a hierarchy of using this. So whenever you learn a language, there is a hierarchy in that. What you have to understand that steps in which the language is originated, and how do we use the rules to pronounce a word or to use letters to use to form a word. The same way for genes also, for identifying the function of the genes also, there is a natural language which is formed in the body. Okay, there is a natural language for this genes. That natural language is actually here used as gene ontology, and this gene ontology can give you the function of the gene. Okay, now how it is done? So people started getting the data for all these things. Okay, so I think if people know certain things like post-translational modifications, there is an activity which is known as phosphorylation, there is an activity which is known as methylation, there is an activity which is known as methylation. Okay, so each activity is done, but when that activity is done, or how that activity is done, and what is the role of that activity being done in the organism. So that is actually a clue for us. And all this information based on the metabolics of the organism, people started understanding the natural language of the genes. That is the function of the genes. And this in detail is known as what? Gene ontology. Now, how does a gene ontology manual refer here? So like any manual here, the language, gene ontology here is also known as a language process, and this actually gene ontology looks for 3 steps, or we can say the vocabulary is actually based on 3. One is it looks what does the molecule do, what is the function of the molecule, first aspect, and second aspect is what is that molecule doing, in which biological process it is participating. Okay, and the third aspect is whether it is a part of any cellular component. So all these 3 give us an information that this gene or protein can have a function. What are the 3? Whether this molecule has a function individually, or whether this molecule is participating in a biological process, or where this molecule is present in the cell, or where is this component present, cell as a cellular component. All these 3 together, they can give us an information that what is the function of the gene. And this people label or name it as gene ontology. And now gene ontology is a big library which was developed, and a vocabulary is being developed on this library, whatever which is identified till date. Okay, based on the molecular function, based on the biological process, and based on the cellular component. Okay, and this is actually a controlled vocabulary. Like any other language, this is a controlled vocabulary which is systematically organized and linked to several things, which is very specific. Okay, it's controlled, it's linked, and it is very specific. Okay, so this is gene ontology. So you can see the example of gene ontology to one thing. Okay, for one thing. Take cytochrome oxidase. I think you people might have learned about the cytochrome oxidase. It is one of the important component which is there in a participating in electron transport. So this you can easily understand what is that. So coming to cytochrome oxidase, C oxidase, the biological process actually, it is the first hierarchy. It's actually a physiological process. Okay, and then what does it do? This physiological process is actually linked to metabolism. Okay, and where it is metabolized, or at what metabolism this cytochrome C oxidase is participating. This is actually participating in electron transport. Okay, so the biological process for cytochrome C oxidase is nothing but electron transport. So this is the hierarchy. So electron transport is a part of metabolism, and that is part of the physiological process, and that is overall is related to biological process. Same way, look at the second one. First one we said biological process, second one is cellular component. So where is this component present? We know clearly it is a part of mitochondria. Okay, so where it is present? It is actually present in the cell, intracellular. And where is that cell? Intracellular component is actually floating in the cytoplasm. All your components will be floating in the cytoplasm. So intracellular cytoplasm is next hierarchy. And where in the cytoplasm this cytochrome C oxidase is actually there in mitochondria. Okay, in each mitochondria it is present. And where it is present in the mitochondria is actually present on the mitochondrial membrane. Okay, and in the mitochondrial membrane, because we say outer membrane, inner membrane, they are there. In where the mitochondrial membrane is present, it is actually present on the mitochondrial inner membrane. Okay, so cytochrome C oxidase. Now we know that the biological process, what it's doing is simply electron transport in the lowest hierarchy, and where it is present as cellular component is mitochondrial inner membrane. Now coming to molecular function, what does it do? It actually catalyzes the reaction. So catalytic activity is the major hierarchy. In that, what is actually doing? We know mitochondria, most of the reactions are oxidation and reduction. So oxidoreductase activity, mitochondria, oxidoreductase activity. Now coming here, what activity it is actually doing? So all the oxidoreductase reactions, they actually require here a heme copper terminal oxidase activity. Or I should say, it is actually cytochrome. Okay, cytochrome is nothing but a porphyrin. So it is a heme copper terminal oxidase activity. Then what is it doing? So oxidase activity, dividing that into two parts, oxidase activity and acting on heme, a group of donors. So heme is there and oxidase activity is performed around that heme. Okay. Now how it is doing? You can see here coming down still, oxidase activity is performed acting on heme, group of donors, and oxygen as the receptor. So oxygen is receptor acting on group of donors, okay, and oxidase activity. So your gene ontology actually is laid like this, with the biological process, cellular component, and molecular function. A detailed hierarchy is there. Okay, this vocabulary, control vocabulary, is now made for each and every proteins, whatever you have. Okay, and people are still developing or improving this vocabulary at this moment. Okay, so now we got clarity. What is gene ontology? Yes or no? Yes. Raise your hands if it is yes. So you don't understand? None of you? Okay, so this gene ontology will tell you actually what is that gene do, or what function does that gene have based on the biological process, based on the cellular component, or based on the molecular function. Okay, so now based on this, we can identify what is the ontology of the gene and see that what is actually the function. So once you get the function, so the top of the hierarchy provides an overview of the function class, whereas the lower level in the hierarchy specifies more precisely the function code. That is what we have seen. The way the protein functionality can be defined in a standardized and ambiguous way. So protein provides three sets of information: gene ontology, biological process, cellular component, and molecular function. So each of which uses a unique set of non-overlapping vocabularies. Okay. So with this, what we can identify is we can identify the function of the protein, the overall functions of the protein. Okay. So the standardization of the names, activities, and associated pathways provides consistency in describing the overall functions and facilitates grouping of proteins related to functions. Okay. So already we have a gene ontology database. So the next way what we can do is whenever we have not identified the function using the blast or motifs or this domains, you can go with gene ontology database. It's already available. So database searching using GO for a particular protein can easily bring up the other proteins related functions in much safer. Okay. So this is your gene ontology. So using gene ontology, a genome annotator can be assigning functional properties of a gene at different hierarchical levels depending how much known about a gene product. Okay, and at present, the gene ontology databases have been developed for a number of model organisms by the International Consortium, in each gene is associated with the hierarchy of geo terms. Okay, and this is where your gene ontology is present. These have greatly facilitated genome annotation efforts. Okay, and gene ontology can be found out at this www.geneontology.org. Okay, I think you don't have a class. Next, continue. You have a class or not?
We have class.
At 10 o'clock.
The class. Yes, sir.
Okay. So I'll just uh, conclude. Okay.
Okay.
This session here. So whenever you have a gene, first you have to predict theoretically, then you have to identify with function, and then we have to go functional assignment. Functional assignment can be done with by blast, by domain, by motif, or lastly by using gene ontology. This is a manual way what you have done till now. All these are manual way of doing. Okay, so there is another way which is known as automated genome annotation. So in the next class, we'll see how we can perform a gene prediction or genome annotation using automated genome annotation. Okay, so on next class on Monday, we'll continue with this automated genome annotation. Is it clear?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
So I'll be ending the class now here. We'll meet in the Monday's class. Okay. Thank you. Bye. And have a nice day.
Thank you, sir.
Thank you, sir.
Good morning, sir. Sir, Balaji, sir.
ఏంట్రా?
Good morning, sir. Balaji, sir.
సార్ గుడ్ మార్నింగ్ సార్. హౌ ఆర్ యూ సార్?
Oh, daddy.
ఏ పిచ్చం వారా?
విష్ణు ఏ గుడ్లు అయినా సరే శనివారం రష్ ఉంటాయి అక్కడ.
Good morning, sir.
Good morning, sir.
Yes. Good morning. Still 10 members are there. So come. 20 is the number including me. Okay, let it be. We'll go with the class. Okay, where we have ended previously. In last class, we have seen about genome annotation, isn't it? In genome annotation, we have seen how a gene can be predicted and how a function can be assigned to a gene. This is what we have learned. Okay, and in that again, we have seen how gene ontology is used to assign a function to a genes. And now, all these are the manual methods, what we have learned previously in the session is how to predict a gene, how to annotate a gene, all of them are manual methods. So, genome annotation now has become automated. There are many tools which have become, which are used here to automatize your data analysis. Okay. So, there are some tools which pick up the genome. If you just give the genome, it will try to say that this is your gene and this is your element, genomic element, and this is the function for this genomic element. This is what the information you get. Okay. So the last week we have seen about manual annotation, genome annotation, and now we'll see how automated genome annotation is there. Okay. So I'll share the screen. Okay, so we'll continue with now with automated genome annotation. Okay, now what is automated genome annotation? So we know that now your genome sequencing has gone a lot. Earlier in 2001, one human genome sequence is available, and later they renamed the project and they said it is 10 genome sequencing projects, human genome sequencing projects. After 3 years or 5 years, they named it as. After that, they named it as 100 genome sequencing projects. The target is get that number. 100 genome sequencing project is to sequence 100 genomes around the world. Okay. Later, it has gone to 10,000, and now it is nearly 1 million genome sequencing project at this moment. Okay, so the target at this moment is to sequence 1 million human genome sequence around the world and understand what is the relationship. Now, the human genome sequencing itself has evolved a lot, that has changed a lot. So they're targeting countries. Now, India itself has sequenced nearly 10,000 healthy individuals and 10,000 diseased individuals last 3 years. Okay, 10,000 healthy individuals and 10,000 diseased individuals, and they started analyzing the data and understand the data. Okay, they started analyzing the data and understand the data. Okay, based on this, people were able to do it. But what is happening while doing all these things? You can see there in 2001, the target is just one human genome, and at this moment, 2021, it's nearly 1 million human genome sequences. So how does it work? In 21 years, the data has increased a lot exponentially. Okay, where is one genome and where is 1 million genome? The data has increased a lot. Okay, so as the data is increasing, we can't go because there for first human genome project, nearly 100 people have come together to just annotate human genome. But now only one person is annotating the same human genome of 10 persons. Okay, so it has changed a lot. In future, one person has to nearly annotate nearly 100 genomes in a month of time. Okay, so the case has said it has become quite opposite the case of 2001 when compared with this situation. So when there is more data which is increasing exponentially, we need to automatize our analysis because we can't sit and do the same old thing. Okay, so we require some automation. So this automation is there, which is required here, and this has led to automated genome annotation. Okay, automated genome annotation. So by this you might have got an idea of why automated genome annotation has been started and why people are going to automated genome annotation. Because there will be 33,000 genes in 3.2 billion base pairs. At the same time, there are 1,20,000 proteins which are there, and we have to coordinate all these things. If we want to do all these things, we can't go manually each and every step. Okay, so this has led to automation of genome annotation. So the other way, what are other possible things that have led to automation of genome? One is huge computational resources which are available for annotating the genome automatically. First thing is that. Okay, so now the data is increased and it is made simple that a simple homology method can be used to detect two gene products which are similar. Okay, and then assign a function to them. That is one. So if manual genome annotation, we have learned that genome annotation, there are two methods. First one is ab initio method, second one is homology method. So based on these two, we are doing it. And the third one has already evolved, that is gene ontology. So all these three have evolved and we are using them. So the first simple method is use homology detection. And this data is available. So as the data is available, we were able to use homology detection to assign a function to genes. Okay, and this is also. So now we use a simple parser. So how many of you have an idea what is a program and what is a parser? All of you, can you on your videos? Yes, on your videos fast. Okay, can anyone tell in the class what is a program and what is a parser? No, I think you have done one paper programming, another DBMS. Two papers you have completed, but this time at least you must have an idea what is that. Don't know?
Sir, actually, this word parcel we are going through for first time, sir. We didn't came across it.
Okay, so tell me what is program then. I'll tell you about what is parser.
Sir, set of
program?
instructions.
Yeah, what is program? Technically, what is program?
Set of instructions.
Set of instructions used to execute something. Isn't it? Set of instructions used to execute something. Okay, so that is the case. Now, with that, people started developing many programs, and many programs are already available to us, isn't it? Now, what is the human psychology? When you have resources which are already available, example, your programs which are already available, do you write again freshly the program or you reuse them?
Reuse, sir.
We reuse. Okay, we reuse them. That is a human psychology, isn't it? In that case, if example for gene prediction, one program is already available. For gene annotation, another program is already available. Okay, and to publish the results of gene annotation online, already a program is already available. So if you want to do genome annotation, that is gene prediction, functional assignment, and publishing it online, do you use what are already available or you just start writing fresh again? What do you do?
We use the available ones, sir.
Okay, we use the available. This is a human psychology. But another problem is there now. Gene prediction is available separately. Gene annotation is available separately. And publishing the results is available separately online. These three are three different programs. They are available now freely. Now, what should we do? Should we combine all these three? Or again write a new one?
We can combine.
We can combine. Okay. So what you're doing? Combining means already one program is there, gene prediction. Second program is there, gene annotation. Third program is there, publishing online the results. I mean, whatever you have predicted and linked function, that should be published online. Then only your data is correct. Okay, everything is done. If you don't publish anywhere, either in the paper or online in a database, that means your work is not completed. Okay, so what are you doing there? You're combining three. What do you do? What do you call when you do this manually? Not programming. When you do that, take three things together and keep it in one way and make it available. What do you do? What do you call that as?
Parcel. Parcel. Sir.
Parcel. The same version of that is known as parsing. Parsing means linking 3 or 4 different objects together and make it available as single object.
Sir, could you spell it?
Parse, P-A-R-S-E. Parsing means P-A-R-S-I-N-G. So what you're doing here is you just take all different programs which are already available, combine together. I think you have learned in C that you can open a program, close a program, upload a file to program, and end the program. In file handling, you would have learned this. So now what you do is you just say, you can use the same C and say that open, close, run this program, 3 steps. So your whole gene prediction is done. Again, after finishing that, collect the results in a file, which is known as in file. You know, any output in C will be stored as out file. So that out file is made as in file for the next gene annotation. Again, you say open gene annotation, upload this in output file as here, and then run. Again, close. Then repeat the same thing. Then open this output file as in file to publishing, then run and close. Output file will come. So what you're doing, just you're making a pipeline. Technically, people call this as pipeline. You're piping all the things to a single client. Okay, 3 different pipelines are there. You're connecting all with them with a joint. Maybe you can use whatever joint you want while connecting a pipeline. Here we call it as parsing. Okay, so people started using parsing here to automatize your genome annotation. Okay, people started using parsing for automatizing your genome annotation. So this is what has happened. First step is available of genome data at exponential level. Second step is available of programs already and no need to write second time the program. So use the programs while parsing. Okay, so while using this, automation has increased a lot. Now we can see how we can use it. So this is the first one. So in addition here, what was known to us? Okay. So previously I said that automation has happened because we have already you can do all these things and parsing of the programs available. Okay, and lots of data has already accumulated on the genes. Now, in addition to homology or sequence matching, conserved motifs have already given us a clue. Okay, and now motif and domain tools are more in number now. So earlier we used to have only one motif database and motif searching tool. Now we have some 10 to 11. At the same time, earlier we used to have only one domain database or more domain search tool. But now we have more. So what has happened is all more tools in addition has made us to think automatic. So we use the first tool. If you found the function, okay, take the function. If not, take the second one. So what we do is we run all the 10 tools and then see all the 10 results and see what is common result among the 10 and take that as your result. Okay, and this has also helped genome annotation automatize. Okay, different tools have made us think that run all the tools and get the common result and parse them to your annotation. Okay, so while doing this, what has happened? Error has come down, procedure has become automatized, manual thing has come down. Okay, and we have multiple evidences, and we have multiple evidences before assigning a function to him, and this is actually minimizing the errors. Okay, so we have different methods. We have minimized the manual process, and we have multiple evidences for assigning the function to a gene, and this has actually minimized errors. Okay, so this is actually a tool which is already available. Okay, for genome annotation, automatized genome annotation. Okay, so the following algorithm is an example that goes a step beyond examining sequencing that and provides functional annotations based on multiple protein characteristics. Okay, so multiple protein characteristics are identified, and then it gives a function. So the tool here is GeneQuiz, and it is available at this. It is available at EBI. Okay, so it is actually a protein sequence annotation thing. So what does it do is this program compares the query sequence against database using BLAST and FASTA. BLAST is one homology similar tool, and FASTA is also a homology similar tool. Okay, it runs both of them, it identifies the homologs, and then it gives you the combined result of both of them. So again, in addition, after doing the BLAST and FASTA, it goes through ProSite and blocks for domain analysis. Okay, it goes through ProSite and blocks for domain analysis. At the same time, it looks for secondary structure analysis. Okay, secondary structure analysis. So what does it look there? The second structure, we have coils, we have transmembranous helices, we have loops, okay, and we have a lot of things. I think you people have learned what are all there which are present in a secondary structure. Can anyone tell what are the secondary structures and what are super secondary structures of any biomolecule? Please on your videos. On your videos. Srividya, what are the secondary structures of a protein? Can anyone tell? Srividya, or anyone you have to tell? Others also on your videos? What are the secondary structures? Srividya, you're still mute.
Sir, plate-like sheets.
Platelets?
Sheets, sir.
Platelets, sir.
Not platelets, sir. Sheets, sir. Beta
what sheets?
plated.
Okay, pleated sheets. Okay, one. What is other secondary structure then? Yes, all of you on your videos. Meanwhile. Can anyone tell? Those who know, raise your hands.
Alpha helices.
Oh, okay. Those who know, raise your hands. I'll ask them. Don't answer, all of you. All of you on your videos first. Okay. Tell me what is another structure.
Sir, alpha helices, sir.
Okay, alpha helices. Okay. You just, right? Balaji? What is the other structure?
Sir, alpha helices and beta structures.
That's all. Both only two. Never
Sir,
there will be two only in the universe. What are things? This is over in the biomolecules paper. I think actually madam has taken you the biomolecules paper, isn't it? Oh, yes. Balaji, who has taken biomolecules to you?
Sir,
I should confirm.
Hari Lakshmi ma'am, sir.
No one is there, sir. Biomolecules, I'm asking.
Yes, sir. Hari Lakshmi ma'am in first semester. Research scholar.
Okay. But I don't think the complete paper is taken by her. Okay. So what are the other
Sir.
structures then?
Sir.
Alpha helices, beta sheets. Only them.
Yes, sir.
Yes, sir. Yes, sir.
Alpha helices, beta sheets, turns, coils, loops. Okay. Alpha helices, beta sheets, turns, coils, loops. Okay. These are the secondary structures. And there are more super secondary structures. We'll leave them. More super secondary structures are also there. But we'll leave them there. Just remember these are the secondary structures. But the question here is now, what is the importance of the secondary structure? Who knows the answer? Raise your hands. What is the importance of the secondary structure? Yeah, Balaji, tell me.
Sir, like due to the second structure, we can determine whether the protein can interact with other, like whether it can interact or not.
Oh, it's not that. There is more importance. Okay. Anyway, I'll be telling you this in the fourth chapter, the relationship between the primary, secondary, tertiary, and quaternary structure. Relationship between this. But I'll just give you a line here because there I'll be telling you in detail. The thing is, your amino acids will decide what is the primary structure. Your primary structure will decide what secondary structure patterns can be formed. Your secondary structure will decide what super secondary structures can be formed. Your super secondary structures will decide what tertiary structure you will get. Your tertiary structure will decide what function it has to do. Okay. So I think this is actually based on an algorithm which you already might have learned in computational biology. Have you learned about hidden Markov model? Hidden Markov model. No? So go back and refer that. I'll just give you a line here. Hidden Markov model is that which says that what is the first event and what hidden information it has for the last event. They are all chain of reactions. For example, you take a primary structure of the protein. I said that amino acids will decide what primary structure you'll get. So first information will decide what is the second information. This event will decide what is going to happen the next. Okay? And amino acids which are there in the primary sequence, that actually decides your secondary structure. So your primary structure is actually deciding what is the secondary structure. So that is actually an event which is governing the next event. And your secondary structure will decide what super secondary structures to be formed. So that event is actually deciding the next event. So if you look like that, come back, the first event is actually dividing the last event. So all of them are in a chain. All of the reactions which are happening in nature are in chain. And hidden Markov model has explained this clearly, and we are using hidden Markov model to see what hidden data is there in the first information to get the final information. Okay, and this is what which is there. So your amino acids will decide ultimately what structure and what function a protein will do. Clear? So this is important. So that means there is an importance between the secondary structure which is formed and the final structure and function of a protein. This is what we will decide. Okay, so that is the importance of secondary structure. And we have alpha helices. Helices are of two types, alpha and beta. But alpha are predominant. Again, beta pleated sheets. Pleated sheets are again of two types, alpha and beta. But here beta are predominant. We have turns, we have loops, we have coils. So how many have said? Simple structures are 5. Super secondary structures I'm not telling you. They are later, we'll see in later classes. Okay. So with this importance of secondary structure, now what you can decide? You can decide what is actually there with your protein. So there is a lots of information which is available. Okay. First thing is we have blast and FASTA, which look what two sequences are similar. Then we have ProSite and blocks, which look at the domains. Then we have several secondary structure analysis tools, which give us the information of the secondary structure. Okay, and then the other things is, now what we do is we take the sequence similarity, we take the domain analysis, we take your secondary structure analysis and others also, and then see what is the result here. So automated gene addition here, genome annotation with gene quiz looks at all these things, and then it assigns a function with the confidence level which is clear or tentative or marginal or negligible. Okay, the confidence levels are these: clear, tentative, marginal or negligible. Clear means the function is 100% true, we can say. Tentative means the function here is supposed to be a function. We have to confirm it again. Marginal means this can be the function. Negligible. Prediction has been done, but we can't take into account. Okay. So these are the things what we get with automated gene annotation with your gene quiz. Okay. And another one, what should I say? Hypothetical proteins. This is a major result to see when you go online to the databases. This is a major function what you see for most of the proteins. Can anyone tell what is hypothetical protein? Just come online. Just come online, all of you. What is hypothetical protein? Anyone? You know, raise your hands. Only one more. Okay. What I'll do is with this question, I'll stop here. Again, we'll meet at 10:35. Okay, and there we'll continue with this question. And meanwhile, if you can gather information, gather what is hypothetical protein, because this is the most important. And I think you people already knew it because practical is already over. There you might have got functions for gene prediction when you've done. Most of the proteins are hypothetical. So by this time you should know what is hypothetical protein and what is the importance of that. Okay, so we'll meet at 10:35 sharp. Okay, okay, then we'll take a break. Yes, all of you on your videos. Okay. So the question what I asked you before the break was, what are hypothetical proteins? All of you on your videos still. Okay, Gunadeepak, Balaji only 2. Rest of you don't know. Okay, Gopal Rao.
Sir,
Tell me what is hypothetical protein?
sir, the hypothetical protein is a protein which existence is known, sir, but its function is unknown.
Yes, its existence is known, but its function is unknown. Okay, So what we are doing is, while doing gene prediction, okay, down your hands now, all of you. So we might have done gene prediction already. What do? We say that this is the gene, and we can use ORF to translate that gene, but we get a protein sequence. So what is happening here is, when we use these tools, all different tools to identify the function of that gene, we were not able to get any function to them. But we definitely know that that is a protein. So what people started calling is that gene, that protein is existing, but we don't know what is the function of that protein. Okay, and there is another one also which is similar to hypothetical, which is known as putative. What do you mean by putative? It's also same. Supposed to be. This is supposed to be a protein. Putative means supposed to be. This is supposed to be a protein, but we don't know what protein it is. So some people, whenever a person has gone through these tools, all these tools, they've identified a protein, they gave a function to a protein. Okay, it's fine. But wherever we are not able to identify the function of a protein, people started labeling them as hypothetical proteins or putative functions. Okay, and now, even now, before doing an analysis, we have to see that all the putative proteins or all the hypothetical proteins are also labeled with a function. Now that itself is also a major function. Okay, people started looking at new tools or new methods and new ways of identifying what can be the function of these putative or hypothetical proteins. Okay, so now you understood what is putative protein or hypothetical protein. These two names are used as synonyms for the proteins where functions are not identified. Okay, and databases are also now have given another way. If they're not able to identify the function, in that there is a domain. They have identified this part is actually a domain, or this part is actually a motif. So for that, they gave a name also again, domain unidentified, DUF. Okay, for that also there is a number, DUF1, DUF2, DUF3. Like that, the number is there actually, domain unidentified. Okay, so you have to remember these three terms, that what is a hypothetical protein, what is a putative protein, and what are the domains which are not identified, the DUF, role of DUF. Okay, so now we'll see what is. So after identifying the function, either using manual annotation or automated annotation, still 40% of the genes which are sequenced are not known to us, and they are named as hypothetical proteins. And now what we can do is, we have to go through experimental methodology to identify them. Okay, experimental methodology. But even this experimental methodology is also time consuming, and sometimes we may not get the function. Okay, so experimental discovery of the functions of these genes and proteins is often time consuming and difficult because lack of hypothesis to design experiments. So now we don't have any rules or to say that, okay, we have a hypothetical protein, follow these rules in the wet bench and identify the function. There is no standard rule or hard and fast rule to say that do this experiment and you'll get the function of this protein. Okay, so that is not there. So in this situation, what we can do? So in this situation, what we can do is, we can predict the function of this hypothetical proteins by searching for remote homologs. What do you mean by remote homologs here? Okay, remote homologs. What do you mean by that? Okay, so remote homologs is one way is we can combine motif information, domain information, secondary and tertiary structure information. So when you search, it gives you some information. That is, you predict some motif. It says the motif is there, but we don't know what is the function of motif. Then you predict a domain, and we see there, but we don't know what is the function of a domain. At the same time, we can predict a structure, primary structure, secondary structure, tertiary structure, quaternary structure. but we don't know what are there, then they're seen individually. So combine all these information, but not in the same organism. Okay, combine this information wherever you find. Okay, combine this information wherever you find. Now, what do you mean by remove? Okay, a question here is, all of you on your videos now. I think you people have performed blast, isn't it? All of you on your videos? I think you people have performed blast. Yes or no?
Yes, sir.
Yes, sir.
So when you perform the blast, I'll ask one. Akhil. So when you perform blast, did you get the results of only one organism, or did you got results of all organisms which is similar across phyla? Across phyla means right from bacteria to mammals, advanced mammals. What is the results? How did you get them?
We can get all the organisms, sir. But when we specified, we can get that specified.
Okay. So you can get results right from bacteria to human or mammals who are advanced, isn't it? While doing blast.
Yes, sir.
So here remote homologs means you have identified a domain which is similar to our domain somewhere in another animal, just not related. So commonly what we do is we look for bacteria, means you're taking bacteria sequences, you look only for bacteria. When you're taking a fungi sequence, means you look only for fungi. Or when you take a human sequence, you look only for human or mammals maximum. But here remote means take a bacterial sequence if you want and try to search across all the phyla. Okay, you look for homologs which are present across all the phyla, but we carefully evaluate. We just can't blindly say, okay, these two are similar, so it's similar. We have to carefully evaluate in which phyla it is present and how it is present. So we look for similar domains, similar homologs, similar motifs, similar primary structures, secondary structure, tertiary structure, quaternary structure, and then combine all these results and see whether it makes any sense or not. Okay, so that is the meaning of remote searching for remote homolog. We try to search for all. That means we are doing deep search. Okay, in other words, you can say deep sea search. Deep sea search indirectly. So you're making a very wide search, and then we identify any domains, any motifs, any primary structures, secondary structures, tertiary structures, quaternary structures, any homologs. Then combine all this information of all these remote homologs, then see what is it's making sense, what sense it's making from this data, and then we can say that this is a function. Okay, and in this way, now people are trying to identify new domains, new motifs, new genes, or new proteins with new functions at this moment. Okay, so this is another study what we're trying to do. So now we understood what is remote homolog, all of you? Yes or no?
Sir.
Okay, so after performing this remote homolog, then we can get the information. Okay, so sometimes what we're doing, we are using this tool, I said, hidden mark model, HMM based. So HMM is actually a tool here which, based on the primary information, it can predict the next information, what is actually there. Okay, so whatever information is set in the DNA sequence can actually give. So based on this line only, the whole bioinformatics work and predictions are done. So there is always a hidden information in your DNA. So whatever hidden information is present in our DNA, we're trying to extract it and understand it. So hidden marker model actually based on motif and domain is smart here. Okay, smart and interpro. Smart and interpro are hidden marker model based tools. Okay, another one is also profile tool. So what are we doing for? This is for functional, similar homologs or domain or motif. But how do we identify a structure or function or a primary structure, secondary structure, tertiary structure and quaternary structure. Okay, so there are some methods which are there, which are known for us, structure-based approaches, which can be used for identifying the tertiary structure. Okay, those are nothing but threading and fold recognition. Even threading and fold recognition are used here for identifying the function of a protein. Okay, so what do you mean by thread and what do you mean by fold? Anyone? Any idea? Do you know or do you don't know?
Sir, could you repeat?
Yeah, Balaji?
Sir, could you repeat, sir?
Okay, you don't know.
Sir, can you repeat the question, sir?
What is threading or what is fold recognition? Yes. You don't know?
No, sir.
Okay. So then I'll tell you in detail in the fourth chapter what is threading and fold recognition. Okay. At this moment, remember that threading and fold recognition are two different methods to identify or predict the secondary structure. Sorry, it's not secondary structure, the tertiary structure of a protein. Okay. So threading, fold recognition, and your homology based method. All these three together, we try to identify the tertiary structure of a protein. Remember that in detail we learn in fourth chapter. Okay, so we can use different structural based approaches such as threading and fold recognition for predicting the. Okay, and now is another way. Still, at least now we have seen that homologs are there, and now we have seen a protein domain can be used, motif can be used, or structure-based approaches, fold recognition, or threading can be used. But if these also fail, then what should we do? Okay, if this also fail, what should we do? So that is what we are going to see now. The distant homologs detected using the structural approach, okay, are linked with well-defined functions, a broad functional class of the query protein. If not, a precise function of protein can be inferred. Okay, that is what we said with structural approaches. But if these also fail, then we have another method which is known as know the subcellular localization. Okay, know the subcellular localization, where this protein is present in a cell. In a cell where this protein is present. That is another one. And based on the subcellular localization, you can also predict what is the function of a protein. And now, even if this fails, subcellular localization, then another one is we look at protein-protein interaction for further functional hints. Okay, protein-protein interaction. I think you people have already done protein-protein interaction, which is there using string, isn't it? You have done using string. Okay, that string is actually again one of the sort of getting or identifying or predicting or getting a hint for function of a protein. Okay, so the protein-protein interaction may also tell us what is the function of a protein. Okay, so we can use all these homolog search, domain and motif search, then structure-based approaches, then subcellular localization, or then protein-protein interaction for a hypothetical protein or a protective protein, and then know what is the function of the protein. If we get any clue, then we can use these clues to design a wet lab experiment and then confirm that information in a wet lab. Okay, so these suggestions do not guarantee to provide correct annotations for hypothesis, but may provide critical hypothesis of a protein function that can be tested in the laboratory. So this gives us a clue. So this remote homology detection helps us to shed light on the possible functions of the proteins that previously have no functional information at all. Okay, so previously we don't have any functional information, but now by doing this homology search at remote and domain and motif search and then structural approaches like threading and fold recognition, then subcellular localization or protein-protein interaction. All this will give us the information. This can be the function of a protein. And then we design a new experiment and confirm it in the lab. And this is how a putative protein or hypothetical protein function can be known. Okay, so that's the mathematics analysis can spur an important advanced knowledge in many cases. Okay, but some hypothetical proteins, because of their novel structural force, still cannot be predicted even with these advanced mathematics approaches and remain challenges for both experimentation and computational work. Okay, and still this is a challenge for us. Okay, so what did we learn till date? We learned that the steps in genome annotation, that is function, gene prediction, and functional assignment is what we have learned first step in manual. And the second step in manual is go through gene ontology and assign a function to thing. And then we have learned about automated genome annotation and how gene twist works. And also we have learned annotation of hypothetical proteins. What are the steps which are involved with annotation of hypothetical proteins? Okay. So this is what we have learned in this genome annotation. So any questions till now?
No, sir.
No?
No,
So people have understood what is genome annotation? Yes or no?
Yes, sir.
See, one question, because I think people know how your second mid exam will be as per the academic calendar.
Open book.
So what do you understand what is an open book, all of you?
Yes, sir.
What is that?
So
Okay,
we'll be given questions, and there will be no invigilation. We need to go through the sources we have, and they have to write the answer.
Yes. So whatever syllabus is there, right from this genome annotation till the next mid, all that syllabus will only be in open book. Okay. So be prepared, and each one will get your own question only, not the same question to all. Clear? All of you.
Yes.
All the questions will be, you have to search and tell me, but each one will get your own question and you have to search. So from this topic, think both theoretically and practically what you can do for your exam. This whole thing will be in open book. Clear?
Sir, what will be the pattern, sir?
That one day before only I can tell you what is the pattern and everything.
Sir, like we need to write it or MCQs?
See, open book means there will be no MCQs. You
Okay, sir.
have to write on your own. Okay? Clear?
Yes, sir.
And one thing is, best of two means here, first and third one will be MCQs and second one will be open book. So open book, definitely one open book exam will be considered for final, and best of two, either one or three will be chosen for your final exams. Internal best. Okay? Be clear with this aspect. So question will be given and you have to answer the question. And only one question will be given to you All of you answer that question. That's all. The same question will be split into several marks. Clear? All of you.
Yes, sir.
Yes, sir.
It should be a theoretical and practical aspect of the question, not theory or practical. It should be mixed. I'm very sure that I'll give you that question. But only one day before only I can tell you in what possible way I can give you a question. Clear?
Yes, sir.
Okay then. So with this genome annotation, we have completed the unit 1. Okay. So tomorrow's class, from this week onwards, all the 4 classes I'll be taking for genomics and proteomics. Okay. So tomorrow's class I'll be coming. Is it clear to you?
Okay, sir.
Okay.
All of you.
Yes, sir.
From tomorrow,
Sir.
all the 4 classes from today onwards, for all the 4 classes I'll be taking. So Deepthi madam will not be coming to you from today onwards. Is it clear for this theory?
Yes, sir.
Yes, sir.
For lab?
Okay, sir.
Lab madam will continue. Theory I'll be taking completely from today
Okay,
onwards.
Okay, sir.
So again, we'll meet in tomorrow's class. Okay, then bye. Thank you. And have a nice day.
Thank you, sir.
Thank you.